So I just wanted to, so the title of the talk today, is part of it is this, and also another chapter of my dissertation that I like talking about. But to kind of get you sort of oriented of what I do. I keep doing the wrong one. There's this long-held view that that genome size in plants can impact a phenotype through direct effects on cell size. And so in other words, if you have more DNA content, you produce bigger cells. And so we might be able to see this sort of manifest itself at higher phenotypic scales. So here we see a plot of allium, or onions, and vicia, which is a type of the legume. And we see that we might expect that as genome size increases, we would expect larger seeds. But in some ways, this is an anecdote, right? This is like Uncle Joe smokes, and he didn't die from cancer, right? But if we sort of look at a much broader assemblage of species, we see that the relationship is much more complicated. So here's the same DNA content and seed mass relationship, and we see that there are some parts of this distribution where we see this positive relationship that we saw in the much smaller study. We also have relationships where it's actually negative. So in some cases, the relationship is clearly much more complicated. But that might not always be the case. We might, as we extend out to more species, we actually might see universal trends. And in this case, we confirm that DNA content does, in fact, impact cell size. So basically, my research is essentially not studying whether or not Uncle Joe smokes. It's actually looking at broad assemblages of species and trying to extract broad patterns and new trends from that data. And so today I'm going to talk about two papers that we've published on this topic, and it's mostly going to be discussing the evolution of a binary character, or characters that are the presence of absence of some certain feature. And I've also developed methods that, as Brian sort of alluded to, that allows for heterogeneity in this process across large species to be taken into account.
Jeremy?
Yeah.
Can you tell me what the differences is between these slides and the previous one? It looks like these are particular cells.
Yeah, this is guard cell size and epidermal cell size.
And the ones before this?
Or seed mass.
Ah.
Yeah.
Okay. So,
Yeah.
completely.
Yeah, the point is that if well, failed that one. All right. No. Great. Confidence is shaken.
It's my fault. I can't read the bloody slide.
All right. Hi, I'm Jeremy. Welcome. So our view of relationships within flowering plants has improved a great deal over the last decade, thanks to dedicated efforts of resolving the tree of life. And so now we recognize the certain clades as representing the beginning of important histories, such as favids, malvids, lamiids. And each one of these groups have given rise to a huge amount of the plant biodiversity. Oh, gosh. And the group that I study is no exception. The campanilids, they actually represent 35,000 species, and they've given rise to several important groups. For example, we have the campanilids include the aquifoliales, which is the hollies. It includes the asterales, which includes the bellflowers and the composites. You have the apiales, which includes the carrots and ginseng. And finally, dipsicales, which is the group that contains viburnum and honeysuckles, very common plants. And there's been this dedicated effort to resolve the relationships within this group, and this is the result. Now, while these studies are very important for deciphering broad-scale relationships among these species, they're not so useful in a comparative context. Because what we can really do with these sort of things is only put characters at the tips of these, where these characters don't change much. Because each one of these tips actually represents a lot of species. For example, this tip actually could represent 200 species, or this one 10,000. So we really can only sort of look at characters on this end of the spectrum, things that are more conserved because we can score those with confidence. So we really don't know much about those sort of labile aspect of trait evolution. Ideally, we'd want a tree that has better representative of the diversity of the group. And so for the past few years, I've been involved with several methods that produce large trees. And the one approach that I'm going to talk about today is the approach we call flawed. It means phylogenetic assembly with databases. And it's kind of a simple workflow. You identify a gene of interest for a clade of interest. It scrubs all the DNA, or it takes all the DNA from GenBank, and then puts it through this rigorous test. One step is to make sure all the sequences are unique. Another is to sort of align each one of align things in an informative way so we can maximize information within the matrix. And we keep repeating the process for all the particular genes we have of interest, concatenate them into a big sparse matrix, and we can use new developments in tree inference to handle this amount of data. And so here's my tree of companionlids, which is 8,900 species or so. And now this represents 30% of the diversity in companionlids, which is a huge improvement from what we used to do before, which we have trees of the same group with very, very, very few taxa. So in a comparative context, we can start to better examine characters across large scales. You good, Lou? See it? All right. So going back to the idea of looking at more labile characters, a really good one is growth habit. Growth habit is one of those characters that really we haven't studied at large scales because there has been this sort of assumption that they vary too much to really be understood. And a good example is in the Asteraceae, the composites. If any one of you guys have ever been out in the field and saw a composite inflorescence, they're very recognizable. You saw them, you would know you were seeing Asteraceae. But if you went out into the field and all you saw was the growth habit, you wouldn't be able to know what it was, because in Asteraceae, growth habit varies a ton. You could have things that are woody trees that are actually really closely related to things that are annuals and herbaceous. So again, it just shows that within this group, growth form is highly labile. But if you step back for a second, and if anybody knows anything about botany, you might think of a few exceptions to this rule. And a couple that come to my mind are things like the oaks, like the Fagaceae. It's a group of woody plants, and they seem to, everybody that's all the species that are in existence today are woody, and it seems like their entire history they've been woody. Then you have groups like the Brassicaceae or the mustards. They have had a history of being predominantly herbaceous. So while we see clades like Asteraceae as being highly labile, there's also a lot of groups where we don't see that many changes in growth habit. So what this sort of comes down to is that the growth habit or binary characters actually might evolve at different rates and different paces in different parts of the tree. But for decades, a lot of our models in comparative biology have assumed this homogeneous process through both time and across taxa. But it's becoming increasingly recognized that there are underlying factors that can affect the phenotype of a clade or a character and will manifest itself as differences in the rates of evolution across the tree. And so I've dealt with this in a couple ways. One, which Brian alluded to, was this using the Ornstein-Uhlenbeck process, or OU, we can refer to it, and where you can actually apply complicated models that allow you to sort of look at whether or not selection or drift dynamics is affecting the phenotype, either across differences in clades, which is highlighted here. We can do it across pieces of branches or branches. And so it's a really, really useful tool for understanding heterogeneity in characters. But for binary characters, there hasn't been much development in terms of heterogeneity. And so here is my companionlid tree, and I scored all the species in this tree for either being woody or herbaceous. And if we did the usual statistical test that people usually do with this sort of data, there's really two models that we would do. One is this one-rate transition model where lineages that are going to transition from woody to herbaceous or herbaceous to woody, that happens at a rate that's equivalent. So going from herbaceous to woody or woody to herbaceous happens at the same rate. An alternative model would be to allow those rates to vary. So in this particular case, we find that going from woody to herbaceous is less likely than the reverse. And what we can do is compare these models and compare like some statistical measure of goodness of fit. And here we use AIC. And we find that this two-rate model is the best fit by a large amount here. But a good question is, are we done? In some ways, it seems really unsatisfying to have 9,000 species and only fit a model with 2 parameters. It just seems really useless. So we decided to develop a model that's inspired by something that's called the Covarion model, which was used back in the 90s to infer phylogenies from sequence data. And the basic idea is that if you see a transition from A to T to C, under the Covarion model, each one of these nucleotides they're either be turned on, where we assume that transitions can happen, or they're turned off, where we assume no evolution. And so what we have to do is, in order for a nucleotide to change, that nucleotide has to be turned on. And so here we see that in this case it's off until it's turned on before we turn to C. So under this model, we assume that each one of these observed states is ambiguous for the different rate classes. So each one of these observed states are coded as being on or off. And then we let the model and a transition matrix to infer where these sites are changing. So our model, which is a little slightly different but inspired by the Covarion model, we refer to as a hidden rates model. And again, instead of assuming that there are two rate classes of being on or off, we assume now that the rates are either fast or slow. And just like the Covarion model, we assume each observed state is ambiguous for the rate class. So we can only observe states, we can't observe rates. And so we let the model sort of tell us where these different rate classes are occurring in the tree. And so this is a method that we just recently published in Systematic Biology. And again, this is also what the rate classes here are considered hidden states in the model. So we fit that model to the same data set, and we see that the transition between woody and herbaceousness is complicated. And here in the upper slow category, we find the same model that we saw before with the two rates, which is that going from a woody to herbaceous is less likely than going from herbaceous back to woody. But in the fast category, we find the exact opposite. We find that in certain parts of the tree, that going from woody to herbaceous happens more often in the fast category than the reverse. Now we can compare the fit of this model with the other two models we discussed, and we see that just having a single hidden rate class or additional rate class improves the fit of our inference by 400 log likelihood units, which is an extraordinary improvement. But why we need not stop at just two categories. We can have as many rate categories as there are branches in a tree. And so, for example, we could have a hidden rates model with three categories, where they can represent slow, medium, fast. And we can just keep fitting more and more complicated models until the information in the data is exhausted. And here we see that with the three category, which is our best fit model, actually improves the fit from the two hidden rate model by an additional 100 log likelihood units. And this was the best model overall when we fit to the data. And if we take a look at the transition, again, we see it it's even more complicated than before, that you see this sort of gradient of increasing rates as you go from slow to fast. And we also find that in the woody slow category, that this woody slow category is actually an absorbing state, meaning that things that move into that state aren't likely to move out, which means they might be stuck in that particular state. But it's important at this point to sort of reflect on what these hidden rates actually represent. And in our view, they represent an unmeasured factor. So it could be that in our woody slow category, which is an absorbing state, that could represent a loss of a gene, for example, where the gene that allows you to go back to being herbaceous has been lost completely. Or it could be that these woody slow taxa are in places in which being woody is selectively maintained, right? So we just assume that these represent some unmeasured factor. But what a really cool aspect of this model is that we can actually paint where in the tree these transitions happen, and where we might expect transition to happen more frequently than in other parts of the tree. So looking at our woody slow category, here we see this is the parts of the tree where we expect things to be woody and unlikely to change. And actually, in a lot of these cases, they make a lot of sense. So here, in particular, we have the Aquifoliaceae, which is the hollies, which a lot of you have seen. They're usually really tall or shrubby, woody things, and they're all that way. And so our model sort of infers that this part of the tree doesn't change their growth habit very much. But if we look at where these elevated rates might be, we see that they tend to be grouping in one part of the tree. And that actually corresponds to the origin of the Asteraceae, or the composites. So again, the model sort of makes sense with our intuition about the group, that Asteraceae, which any botanist would tell you, actually changes their growth form quite a bit. And the model is telling us that. So for this part, I just, the evolution of growth habit in companionlids is highly labile, but it clearly varies by where what group you're talking about, what parts of the tree you're talking about. And it suggests that there are important differences in the factors that limited or promoted transitions in certain regions of the tree. And like I showed before, you have clades like aquifoliales where you aren't expected to see much growth form evolution, whereas Asteraceae, we see lots of change. And just a small plug is that a lot of the work that I said before is looking at heterogeneity across the tree, and with binary characters now we can do that, and we can do it pretty well, and it seems to be working fairly well. And also just to plug, if anybody's interested, it's available on Crayon. And I should also say too that the models that we're showing here are the most complicated for the particular rate class, but you could easily devise more simple or different combinations of character and rates to and fit different models that way. So if you just were looking at the 2-rate category, there's 4,000 or so possible models that you could construct. So it's a really interesting way of modeling this stuff. So some future extensions. One is correlations among multiple binary characters. So if you had, I just showed woody versus herbaceous, but if you had some other binary character, like deciduousness and not being deciduous, you can actually look at the correlation between those two characters, and you can actually use a hidden rates model and see where that correlation might be changing in different parts of the tree. We're also discussing ways in which we can take into account both transitions in the states as well as diversification, which I'll talk about more in a little bit. And then finally, we're working on identifying these hidden rates or hidden states in contemporary species. And so I'm going to show just some basic results that were just kind of finished just the other day. Like I said before, we treat each observed state as being ambiguous for the different rate classes. So here we have a model where we have a slow and fast category. And so we have our woody here, which is ambiguous. We have a probability of 1 of being in either rate class. And what we can do is we can actually infer what the likeliest rate is for this particular tip, using the same methodology that we would as we do for internal nodes. And how we do this is really fairly simple. It involves two passes on a tree. The first pass is where we calculate the conditional likelihood of observing the states that descended from each node. And so we can do that for each node, keep track of that as we work down towards the root. Then the up pass just simply calculates the probability that given this focal node, what's the probability that given this ancestral state, what's the probability of change that's occurring along this branch? We can multiply that probability by the conditional likelihood, and that's how you get the ancestral state at this node. But most people just stop at the internal nodes because usually we know what the state is when we go into these analyses. But now we don't know the states, and we can just continue up and give an estimate of what the likeliest rate and state combination is for each tip. And so what we did is we took that estimate for each one of these tips, and we matched it up to our Q matrix, and we basically made a data sheet that has these rates for each one of the species. And so what we did was, to test out these ideas of estimating rates for contemporary species, is we took a data set that I've been involved in assembling with a nascent working group, which we're called the tempo and mode of plant trait evolution. And the idea is to assemble a database of functional traits for all plants that have some sort of GenBank ID, so that we can match it up with a large tree. And so in this particular case, we built a tree for 22,487 species of angiosperms. And we actually have character coding for woody and herbaceous for each one of the tips here. But one small, just minor point is that when you have a big tree of all angiosperms, the definition of woody isn't necessarily simple one. So if monocots don't actually produce true wood. So when I'm talking about across angiosperms as being woody, what I really mean is above ground persistent stems. And so things like palms and screw pines and the monocots, they don't actually produce true wood, but they actually have a persistent above ground stem. And same with cactus. They are technically fleshy stems, but they also have an above ground persistent stem. Now I'm just going to be using woody as a covering term from here on out. So what we did is we decided to take these rate estimates for each one of the tips and then map where in the world these species are found. And so that meant just taking all the records that we had for that matched with the tree from GBIF, which gave us about 35 million occurrences across the world for about 12,834 species. And so what we wanted to do was look at what we might expect hotspots of change or expected change in growth form across the world. So the first thing we looked at was things that are herbaceous now, where would you expect things to become woody in the future based on the model? And here we see that there are certain parts of the world that are really hot for where you might expect changes to herbaceousness, woody. And we see Europe and the Mediterranean area as being really hot spots. Same with Australia and North America, and really just sort of temperate areas seem to be really likely to change their growth form. And the reverse, going from woody to herbaceous, we see that actually more tropical areas, and actually also Australia as being areas where you might expect transitions to occur. This sort of way of looking at things is very much a work in progress. We're still sort of trying to figure out what this all means. In some ways, it's sort of coarse-grained at this point, and they're trying to develop ways so that we can get a better estimate of where these changes are occurring. But the idea of evolutionary derived geographic layers seem like an interesting way of seeing the world. If you have expected changes in the climate, certain areas might be able to cope with that because they are more likely to change their functional traits like growth habit. So, anyway, this is a future extension that you can use for our hidden rates model. The second thing I want to talk about is some work that was recently published that looked at fruit evolution in companionlids. And what's really cool about the fruit type stuff is that with this large comprehensive tree in hand, we have new opportunities to take a fresh look at these long-standing questions. And so fruits are among one of the most characteristic and diverse structural features of angiosperms. For example, they vary a ton, and we all have particular names for them. And botanists have always been interested in sort of correlating whether particular fruit types can have an impact on diversification. So the idea, for example, is that fleshy fruits allows for better dispersal, get you into new places, and that might actually increase your probability of being isolated, which could lead to speciation. So that's been a long-standing question that people have had about fleshy fruits. There's another group of people that have said that if you're wind dispersed, you're more likely to have higher rates of diversification, because with wind, you can also get isolated and disperse long distances. But to the extent that people have actually looked at this correlation, it's mostly confounded by taxonomy. And so if each one of these titles here of really prominent papers on this topic have really just focused on examining just particular families or taxonomic ranks to do this sort of study. And what do I mean by that? Well, what a really common way of looking at this correlation is people would take a family where one clade within that family has about 40 species in one character, and then another clade that has 1,000 species in another character. And so what they do is they would collapse this down into pairs and compare the diversity between the two clades here. And so at best, this really provides us a really coarse-grained analysis, right? We don't really know how many origins there are within this clade. You're just sort of kind of squinting and sort of assuming that everything in here has the same character. But that may not be true. Or at worst, you can compromise statistical power because you maybe only have a few families that you can actually compare. So we really don't know much about this fruit type and diversification correlation, mostly because of the methodology that people use. But we now have comparative methods that consider state changes across the whole tree when assessing dependence of diversification rates on a character state. And so we decided to take a look at this with companionlids. And what we did is we scored companionlids for 3 basic characters. One is whether or not a fruit was dehiscent. So a dehiscent fruit is one that explodes on its own. It has opens up on its own. Fleshy versus dry is another character, which I'm sure you guys all know. And then having a single seed or having more than one seed. Those are the particular characters that we scored. And we found that companionlids actually have really 4 basic common types. One is a berry, which, again, doesn't open, so it's indehiscent. It's fleshy, and it has multiple seeds. A tomato is, again, a really good example of a berry, not a vegetable. A drupe, which is like a berry, but it only has a single seed. All right? And then we have capsules, which are, again, they dehisce, and they have multiple seeds, and they're dry. And finally, the achene, which is single-seeded, indehiscent, and dry. And a good example of achene here is a dandelion, which you guys have probably all seen, or a sunflower seed, which is technically a fruit. We also found 2 additional rare types that seem kind of strange, but don't actually have a name. We call it the one's indehiscent, so it doesn't open. It's dry, but it has multiple seeds, which we really uniquely called IDMs because we're really creative. And then the second one was dehiscent, dry, and single-seeded, so it opens, it's dry, and it only has one seed. And again, we creatively called it DDS. So what we wanted to do was model how transitions in fruit type happen across companionlids. And a really common way that people do is they use what's referred to as a multiple state model, where these are all the possible transitions you'd expect across all these different fruit types. Okay? And it looks very complicated. But this might be too simplistic of a view of how fruit type might actually happen, or how fruit type changes might actually happen. Let me give you an example. Suppose you're trying to understand, trying to model the evolution of going from a berry to an achene. Well, a berry has indehiscent, fleshy, and multiple-seeded traits, whereas an achene has indehiscent, dry, and single-seeded traits. Now, in order for that to happen, you'd have to see 2 changes take place. One, you'd have to reduce to a single seed, or you'd have to lose fleshiness. The other fleshy fruit type, which is a drupe, only involves loss of fleshiness. So there's only one particular step that goes between a drupe to an achene. So the point here is that with the multiple state model, these sort of jumps are all equivalent, even though they don't represent the same number of state changes. So what we did was we devised a new way of looking at how these traits change. We sort of conceived of this 3-dimensional hypercube where we only allow a single state change at a time to occur. So here now, we have our berry to an achene. Now we see our 2-step jump here. In this particular route, you could you go from being multiple-seeded to single-seeded, and then you go to being an achene. Or you can go the opposite direction, where you can lose fleshiness and then reduce to a single seed. So we fit this model to our companionlid data set. And this is what we found. We find generally that transitions from a capsule out, you're actually not expected to go back to a capsule. And actually, let me fast forward here. Let me start over. So our model strongly suggests that the capsules are the ancestral condition within companionlids. And if you look at the transition matrix, we find that the companionlids sort of started in this capsule state and have moved out. And once that state has evolved, or you've changed one particular character within a capsule fruit type, you're unlikely to go back to that. And we also find that when you evolve these fleshy fruits, these happen more recently and usually are derived from capsule-type fruits, which is somewhat consistent with the fossil record, which suggests that actually fleshy fruited things are much more derived. And finally, we find that the most predominant fruit type in companionlids, the achene, seems like there's all roads leading towards that achene fruit type. So if you see here in the box, you see there's all 3 arrows that are moving towards the achene. So it seems like there's a lot of evolution towards that fruit type. But for these types of models, though, having something that's highly prominent or having a predominant character type can actually be somewhat misleading, particularly when you're talking about transitions towards that state. So, for example, what I was showing before, you have asymmetric transition rates that can lead to an overabundance of traits. So in this case, what we find is that it could be that going from 0 to 1 happens much more often than 1 to 0. So you might expect that's why we see a lot of Akene fruits in companionlets. But you can also think that maybe the transition rates are identical. So if you're going from so the transition rates can be identical, but the diversification rates change. So you could have higher speciation rates feeding back into that Akene state. So that's why you see it, this overabundance of characters. Alternatively, these two processes could work in concert, so that you can actually have asymmetric transition rates as well as higher diversification rates that are causing this overabundance of Akenes. And so there's this model that actually takes both these processes into account, which we refer to as the BISI model, which is a binary state speciation extinction model. And what it does is it jointly estimates the probability of speciating or going extinct or transitioning between particular character states. And these are developed by Madison and Fitzstrong. So we fit this model to our companionlet data set, and we find that it's complicated. So if we look at the speciation rate, the Akene, you're more likely to speciate as an Akene than it being not an Akene. But we also find that the extinction rate is also very high if you're in the Akene state versus not. Finally, we also find that going forward to the Akene state actually happens more often than going away from the Akene state. So if we go back to our model, we see that we see both things happening. We see the reason why we have lots of Akenes in companionlets is that we see a lot of transitions to that state, and also when you're in that state, you're likely to speciate a lot. However, we looked at the estimates of these speciation extinction parameters, and we find that they're not independent of one another. And that's represented here by these scatter plots where the relationship, they're really tightly correlated. What this represents is sort of like, when you have these sort of opposing parameters like this, and this is a bit of a technical point, but when you have opposing parameters like this, that you can actually have different combinations that have the same likelihood, right? So, having a speciation rate that's low but a slightly higher extinction rate can have the same effect on the number of characters you'd expect as something up in here in this graph. So they're really not independent of one another. But what we found was that if we transform these into something that's called net diversification rate, so just taking the speciation minus extinction, we can actually tell the two apart much, much better. So from now on, I'm just going to talk about net diversification rate within companionlets. So here's the model. We see that we transition towards that Akene, but you're more likely to have higher net diversification. In fact, it's 3 times higher in Akene lineages. But we decided not to stop there because, as you can see, the distributions of Akene fruits isn't necessarily constant across the tree. It seems to be grouped in different parts. So we decided to subdivide the tree into the 3 main clades that actually represent the most amount of Akene fruits. So we fit the same BISI model to the Asterales, which is shown up here. And we see the same thing that we saw across all companionlets. You have really high net diversification rate compared to non-Akenes, and we see much more likely to transition to an Akene than the reverse. But when we look at the apiales, we start to see that pattern disappears. So we see that there's some evidence for higher transition rates, but we don't see any evidence of higher net diversification. And finally, within the dipsicales, we see the same pattern that we see in the apiales, that there isn't really much difference between whether or not you're Akene or non-Akene in terms of the diversification and transition dynamics. And if we just plot the diversification rate, it clearly shows that there's something interesting going on in Asterales. That's Asterales, the Akene fruit in Asterales is doing something very different than the rest of the clades that have that same fruit type. Which is interesting because a lot of these BISI models are used to identify things called key innovations, right? You evolve this particular character, and it allows you to increase your diversification rate. And so I could have stopped with the BISI model, showing that, oh, look, this Akene fruit is a key innovation within the group. But if you start to subdivide that a little bit, you start to see that maybe being a keen in certain parts of the tree has a different effect than being a keen in another part of the tree. And so what we did was we decided, OK, let's forget about BISI. Let's just identify where we'd expect exceptional radiations in the tree. And does that correspond to any keen origins? And we find out something that's kind of interesting, that we find that in several places, these shifts that are independent of whether or not what character you are, just what the topology is saying, where the shifts in diversification are, they happen not too far away from where these akenes originate. So here in Asteraceae, we see that a couple nodes away, they see these exceptional radiations. And this goes back to this model that we've talked about as a grad student for many years, which is that these diversifications, these effects on diversification, may not coincide with the origins of particular traits, but they are associated with maybe a buildup of particular characters. So what we started to sort of think about is, you might evolve the akene, right? But you might modify it in some way such that you have, you might have better wings to get into the wind, or you might have the right growth form, or you might go into a new biogeographic region that could somehow relate to being an akene and cause your shift in diversification. So the whole point here is that with the akene fruit type, it's complicated, right? It's not that there was one trait or one explanation to explain why there are so many of them. And it depends on what parts of the tree that you're talking about. And so this, so basically, looking at the BISI model is really, it highlights this problem of focusing on characters, like I said. Again, we're focusing too much on which branches are in which character state, and not focusing on the events, like where in that tree that has evolved, and how that evolution of that trait in certain parts of the tree is different, has a different effect than other parts of the tree. And so we're currently developing, or thinking about developing, this hidden Markov model approach that allows us to extend to this BISI framework, where we can be more explicit as to where in the tree we might expect this trait effect on diversification is happening. I just realized I just blew through this. So some final thoughts. I think the study of these very old, really widespread clades holds the promise of greater insights and evolution across different temporal scales. As you can see, as you move out into bigger spaces, you see the world is much more complicated, and we have to develop new methods to deal with this complication. And I think looking at this large scale, also, they're pretty invaluable because they validate new approaches, like the hidden rates model. They allow us to develop interesting models and help evaluate them. They also, looking at these large trees, they help us assess confidence and relationships. And I also think they help us identify where we might need to improve our knowledge. And I think they also unify efforts, and they build a foundation to do new, more promising work. And so acknowledgements, just to thank Michael Donahue, which is my advisor at Yale. Just a small plug, he'll be here in April giving the EEB seminar. It's going to be awesome. He's a really great speaker. So yeah, I hope you guys put that on your calendar. I want to thank Brian O'Meara, who I've worked with quite a bit, actually, he said, as a graduate student. He's always very positive and very honest, which is good. I need that sometimes. And I think Jim Fordyce is also my UTK mentor. I'd like to thank the Donahue Near Lab, and also the Jeff Oliver, who's at Oregon State. He was a postdoc in the Yale lab, and he was always good about making fun of me, but also being very positive about what I was doing. And finally, I want to thank the EEB department for allowing me to hang out with them and feel like I'm a part of it. And also, I want to thank my funding sources, Nimbus, Yale, the NSF, and iPlant. And with that, I can take questions.